Hello students, let's discuss about genetic code and prokaryotic translation. In introduction, the genetic information coded in the DNA is decoded by the process of transcription and translation in a living system. The process of translating a code requires reference of the language of the genetic code. The genetic code being a universal language of the genetic information of all living systems on Earth, it is essential to understand it as the translation of the code by the living machinery manifests it into a physical, tangible characters called phenotypes. Let us first discuss about the properties of genetic code. The genetic code was discovered by Marcel Warren Nirenberg and Hargobind Khorana. It has the following features. First, it is composed of a 4-nucleotide on mRNA, which is A, U, G, and C. Second, it rich in triplets, that is in 3 nucleotides per codon. In the third, there are 64 possible codons. The start codon is AUG, also codes for methionine, and the stop codon UAG, amber, UGA, opal, and UAA, ochre. The other 60 codes represents amino acids. In the fourth property, the code is degenerate, which means there can be more than one codon for an amino acid. In the fifth property, the code is non-overlapping, and in the sixth, AUG, also known as the start codon, defines the starting of translation. In the seventh, the genetic code is read in a commaless manner, and in the eighth property, the code is universal. The degeneracy of the genetic codon can be explained by the Gooble hypothesis proposed by Francis Crick in 1966. Now, let us discuss about Gooble hypothesis in more detail. Gooble hypothesis. In the first point, the first 2 bases of a codon in mRNA always forms strong Watson-Crick base pairs with the corresponding bases of the anticodon in tRNA and confer most of the coding specificity. In the second point of Gooble hypothesis, the first base of some anticodons reading in the 5' to 3' direction, remember that this is paired with the third base of the codon, determines the number of codons read by the given tRNA. When the first base of the anticodon is C or A, binding is specific and only one codon is read by that tRNA. However, when the first base is U or G, binding is less specific and 2 different codons may be read. When inosine, that is I, is the first or Gooble nucleotide of an anticodon, 3 different codons can be read by the tRNA. This is the maximum number of codons that can be recognized by a particular tRNA. In the third point, when an amino acid is specified by several different codons, those codons that differ in either of the first 2 bases requires different tRNAs. In the fourth property of the Gooble hypothesis, a minimum of 32 tRNAs are required to translate all the 61 codons. Let us discuss about translation in prokaryotes in detail now. The translation in prokaryotes starts with a step called tRNA charging. In the first stage of protein synthesis, which takes place in the cytosol, the 20 different amino acids are esterified to their corresponding tRNAs by aminoacyl tRNA synthetase, each of which is specific for one amino acid and one or more corresponding tRNA. In most organisms, there is generally one aminoacyl tRNA synthetase for each amino acid. As noted earlier, for amino acids that have 2 or more corresponding tRNAs, the same aminoacyl tRNA synthetase usually aminoacylates all of them in E. coli. The only exception to this rule is lysine, for which there are 2 aminoacyl tRNA synthetases. The activation reaction occurs in 2 separate steps in the enzyme active site. In the first step, an enzyme bound intermediate aminoacyl adenylate is formed by reaction of ATP and the amino acid at the active site. In this reaction, the carboxyl group of the amino acid is bound in the anhydride linkage with the 5' phosphate group of the AMP with displacement of pyrophosphate. In the second step, the aminoacyl group is transferred from enzyme bound aminoacyl AMP to the corresponding specific tRNA. The course of this second step depends upon the class to which the enzyme belongs. The reason for the mechanistic distinction between the 2 enzyme classes is yet unknown. The resulting ester linkage between the amino acid and the tRNA has a high standard free energy of hydrolysis. The pyrophosphate formed in the activation reaction undergoes hydrolysis to phosphate by inorganic pyrophosphatase. Thus, 2 high energy phosphate bonds are ultimately expanded for each amino acid molecule activated, rendering the overall reaction for amino acid activation essentially irreversible. The process of translation in prokaryotes can be classified into 3 continuous stages named as initiation, elongation, and termination. Now, let us look into the steps of initiation. Although there is only one codon for methionine, that is AUG, there are 2 tRNAs for methionine in all organisms. One tRNA is used exclusively when AUG represents the initiation codon for protein synthesis. The second is used when methionine is added at an internal position in a polypeptide. In bacteria, the 2 separate classes of tRNA specific for methionine are designated tRNA met and tRNA F-met. The starting amino acid residue on the amino terminal end is N-formylmethionine. It enters the ribosome as N-formyl methionine tRNA F-met, which is formed in 2 successive reactions. The initiation of polypeptide synthesis in bacteria requires the 30S ribosomal subunit, which consists of 16S rRNA, the mRNA coding for the polypeptide to be made, the initiating F-met tRNA F-met, a set of 3 proteins called initiation factors IF1, IF2, and IF3, GTP molecules, and 50S ribosomal subunits, as well as magnesium ions. The formation of the initiation complex takes place in 3 steps. First, the 30S ribosomal subunit binds initiation factor 3, which prevents the 30S and 50S subunits from combining prematurely. Binding of the mRNA to the 30S subunit then takes place in such a way that the initiation codon AUG binds to a precise location on the 30S subunit. The initiating AUG is guided to the correct position on the 30S subunit by an initiating signal called the sign-Dalgarno sequence in the mRNA, centered 8 to 13 base pair to the 5' site of the initiation codon. Ribosomes have 2 sites that binds aminoacyl tRNA, the aminoacyl or A site and the peptidyl site or P site. Both the 30S and the 50S subunits contribute to the characteristics of each site. The initiating AUG is positioned in the P site, which is the only site to which F-met tRNA F-met can bind. In the second step of the initiation process, the complex consisting of the 30S subunit, IF3 and mRNA now forms a still larger complex by binding IF2, which already is bound to a GTP and initiating FMET tRNA FMET. The anticodon of this tRNA pairs correctly with the initiation codon in these steps. In the third step, this large complex combines with the 50S ribosomal subunit simultaneously. The GTP molecule bound to the IF2 is hydrolyzed to GTP and phosphate. Initiation factor 3 and IF2 also depart from this ribosome. The second stage of protein synthesis is elongation, the stepwise addition of amino acid to the polypeptide chain. Again, our discussion focuses on bacteria. Elongation requires the initiation complex as described above, the next amino acyl tRNA specified by the next codon on the mRNA, a set of 3 soluble cytosolic proteins called elongation factors, also called EFTU, EFTS and EFG, and there is a requirement of GTP molecule. 3 steps takes place in the addition of each amino acid residue, and the cycle is repeated as many times as there are residues to be added. In the first step of elongation cycle, the next amino acyl tRNA is first bound to a complex of EFTU containing a molecule of GTP. The resulting amino acyl tRNA EFTU GTP complex is then bound to the A site of the 70S initiation complex. The GTP is hydrolyzed and EFTU GTP complex is released from the 70S ribosome and an EFTU GTP complex is regenerated. In the second step, a new peptide bond is formed between the amino acids bound by their tRNAs to the A site and P sites on the ribosome. This occurs by the transfer of the initial N-formylmethionine group from the tRNA to the amino group of the second amino acid now in the A site. The alpha amino group of the amino acid in the A site act as nucleophile, displacing the tRNA in the P site to form the peptide bond. This reaction produces a dipeptidyl tRNA in the A site and the new uncharged deacylated tRNA FMET remaining bound to the P site. The enzymatic activity that catalyzes peptide bond formation has historically been referred to as peptidyl transferase and was widely assumed to be intrinsic to one or more of the proteins in the large subunit of the ribosome. In 1992, Harry Noller and his colleague discovered that this activity was catalyzed not by a protein but by the 23S ribosomal RNA, adding another critical biological function for ribozymes. In the third step of the elongation cycle called translocation, the ribosome moves by the distance of 1 codon towards the 3' end of the mRNA. Because the dipeptidyl tRNA is still attached to the second codon of the mRNA, the movement of the ribosome shifts the dipeptidyl tRNA from A site to the P site, and the deacylated tRNA is released from the initial P site back into the cytosol. The third codon of the mRNA is now in the A site and the second codon in the P site. The shift of the ribosome along the mRNA requires EFG, also called translocase, and the energy provided by hydrolysis of another molecule of GTP. A change in the 3-dimensional conformation of the entire ribosome is believed to take place at this step in order to move the ribosome along the mRNA. In the final stage of translation, called the termination, Elongation continues until the ribosome adds the last amino acid completing the polypeptide coded by the mRNA. Termination, the third stage of polypeptide synthesis, is signaled by one of the three termination codons in the mRNA, which is UAA, UAG, and UAGA, immediately following the last amino acid codon. In bacteria, once a termination codon occupies the ribosomal A site, three termination or release factors, the proteins RF1, RF2, and RF3, contributes to the hydrolysis of the terminal polypeptide tRNA bond and release of the free polypeptide and the last tRNA, now uncharged, from the P site. This is followed by the dissociation of the 70S ribosomes into the 30S and 50S subunits, ready to start a new cycle of polypeptide synthesis. Release factor 1 recognizes the termination codons UAG and UAA, and RF2 recognizes UGA and UAA. Either RF1 or RF2, as appropriate depending on which codon is present, binds at a termination codon and induces peptidyl transferase to transfer the growing peptide chain to a water molecule rather than to another amino acid. The specific function of RF3 has not been firmly established. In eukaryotes, a single release factor called ERF recognizes all three termination codons. In conclusion, transcription and translation are two interlinked and continuous processes in prokaryotes, and both takes place in the cytoplasm of the cell. The process of transcription makes a copy of the genetic information present in the DNA sequence to an mRNA molecule, which is further translated to a functional polypeptide by the process of translation. Translation makes it possible for the genetic information to be expressed in a phenotype.